Well, to describe this, I need to tell you about a few things. First of all, we want to consider a tree as a model, not a mouse model, a mathematical model. A tree is a model. Its parameters are the branch lengths of the tree and the topology of the tree. That's the branching order, so that's like A joining to B, B joining to C. What we want to do is we want to calculate the probability of a model, in this case it's a tree, given some data. In our case this is some sequences with some times, in order so that we can choose the best one or collection of best ones for the data. Also, just to say, in our tree model with our branch lengths, there are several parts which go into that tree model. So there's like models within the model. And one of those models would be something like a clock rate model describing how quick you think the molecular clock is going. So, the Bayesian part, in order to calculate the probability of our tree given our sequences, is done using Bayes' theorem. Now, this is what we want to calculate. We want to calculate the probability of our tree given our sequences, the probability of the model given the data. This is called the posterior, and this is the thing that we want. If we want it to have a high score or a collection of high scores, this is what we're aiming to get. However, we can't calculate this directly. What we can calculate, though, is this quantity, the probability of the data given the model, the probability of the sequences given the tree, which is called the likelihood. What this is what is the probability of seeing these sequences with those particular mutations given a specific tree. And there are several expressions, mathematical expressions, which will allow you to calculate the likelihood of the data given a specific tree. But in order to get this final posterior, we have to have a couple of other quantities as well. We also need to include a thing called the prior. And this is the probability of having that tree in the first place before you've even seen any data. So the priors, these could be completely uninformative. So in effect, if the priors are uninformative, it doesn't even matter about this part. But the priors can help you, if you like, cut out the really stupid things. So an example of an informative prior would be something informative on the clock rate. So, as I said at the beginning, you have an idea for RNA viruses, DNA viruses and bacteria what the clock rate is. In mathematical terms, in principle, the clock rate could be any number between 0 and infinity. But in practice, you know that's not true. You are not going to have 1,000 mutations in a year. That's not right. It's not going to be that. What you might say is actually for the prior for the clock rate, I think it's going to be approximately 5 in 1,000 per site per year, and it's probably going to be a normal distribution, and it might have quite a wide standard deviation of what the acceptable numbers are going to be, but it will be something like that. So in that case, you would include that as the prior, and that will let you cut out the really ridiculous answers. For informative priors, you would normally base them on either some other data or some other idea about biology, like I was describing about the clock rates. The final part of this equation is this bit at the bottom, the probability of the data itself. In fact, because we're always going to be considering the same sequence data, you can just consider this part to be a normalizing constant. You don't actually need to worry about it, because what you're going to do is you're going to try different trees and score the trees according to the sequence data. But the sequence data is going to stay the same. So that part is going to cancel out when you're comparing different trees with different parameters.